of today's lecture is your RNA post-transcriptional and transcriptional modifications. Many of these modifications take place while the mRNA is being transcribed from a gene or a DNA, and some of the changes occur after transcription. Okay, so there are four kinds of transcriptional and post-transcriptional modifications. Number one is your 5' capping. Second is your 3' polyadenylation. Also, third is your splicing. And fourth is your histone mRNA processing. If we see the structure of an mRNA when it's fully produced, so you have this region, that is the main ORF or the protein coding sequence. Okay, if you go towards the 5' end, so that means this end, so this is 5' and this is 3', we say that we are moving upstream. And if we go 3' towards 3', we are saying we'll go downstream. So you have a 5' UTR, we call it untranslated region. This does not translate into a protein. Okay, and then you have a cap. This is your 5' cap. And downstream of this mRNA or the protein coding sequence, you have a sequence called as AAUAA. Okay, that is the sequence that is recognized by the complex, protein complex that is involved in adenylation. Okay, this is called as the polyadenylation signal. And then downstream to this is your poly-A tail. Okay, where around 50 to 250 adenosines are added. Okay, so initially we were talking about capping. So why we call this 5' is because if we see the structure of your what we call as RNA, you have a okay. The primary numbering is in this what you call as nitrogenous base ring. Okay, so that's why here we start with 1', then we start with 2', then we start with 3', start with 4' and 5'. Now in DNA, this is deoxyribose, you have an H here. Oh sorry, deoxyribose, you have an H here. This is your 3' OH, and this is your 5'. You have a phosphate here. So always the phosphate will be at 5' and OH will be at a 3'. So the bond is 5', 3'. So you have a bond 5', 3'. 3 prime. So if replication is to occur, it occurs in 5 prime 3 prime. If transcription is to occur, it occurs in 5 prime 3 prime. Most of the cellular processes related to DNA RNA, they occur in 5 prime 3 prime direction. However, if there is proofreading and some wrong bases incorporated into the DNA, the polymerases move towards 3 prime 5 prime direction. Okay, that's an analogy. And so so what happens here, so then another phosphate of the next nucleotide is attached here. Okay, then you have the CH3, and then you have another OH at 3 prime. This is your 5 prime, this is your 2 prime, and since this is RNA, you have an OH here. In DNA, you have an H here, deoxyribose. This is a ribose sugar, 5 carbon sugar. And then you have another base attached to this. So usually the bonding in mRNA is 5 prime 3 prime. Okay, it's a phosphodiester bond. This is the phosphodiester bond. The bond that connects a base with your sugar, this is a glycosidic bond. Okay, then when A is paired with T or G is paired with C, this double bond is a hydrogen bond, and this triple bond is also a hydrogen bond. Okay, so now to this phosphate, in 5' cap you have a phosphate, you have 3 phosphates. Okay, but this bond that is present here is a 5 prime bond. Usually you have 5 prime 3 prime bond, but here this bond is 5 prime 5 prime. How? This is your CH2, okay, this is your OH, this is your OH, okay, and this is your guanosine residue. Okay, so it will be like this. So this is your 5 prime, this is your 5 prime. So that's why this bond we call as 5 prime bond, right? So this capping is usually in your eukaryotes. However, in bacteria there is a capping of RNA, but there usually they add NAD positive or NADH at the 5 prime end of your messenger RNA. Okay, mitochondrial RNA and chloroplast RNA, they do not have a cap. Okay, it's only in your messenger RNA, right? So now why this boy, our time is running out. Meeting will end in 10 minutes. Upgrade now to remove it. Okay, so now the question arises, why we need capping? Because it adds stability to your mRNA. Okay, it helps in the nuclear transport. Okay, it helps in prevention by degradation by exonucleases. Okay, it helps in promotion of translation, and it helps in 5 prime proximal intron accession. The intron that is at the 5 prime end, okay, it helps in the accession of this intron. Now, what is the process? How this happens is, I'll run this out. So you have an mRNA with do we have colored? Okay, so you have a triphosphate here. Okay, as just we draw, this is your mRNA chain. There's an enzyme called as phosphohydrolase that removes your, this is an alpha carbon, alpha phosphate, beta phosphate and gamma phosphate, that removes this gamma phosphate. So you have a condition like this. So then after that there is an enzyme called as guanylyl transferase that comes. Okay, this guanylyl transferase also has a guanine. Then it has again 3 phosphates. Okay, guanine triphosphate. This is your alpha, this is your beta, this is your gamma. This guanylyl transferase makes a 5'5' bond with this mRNA strand. So you have a condition like this. This comes out. Okay, after this there is an enzyme called as guanylyl 7-methyl transferase. Okay, methyl transferase that adds a methyl group at this 7th carbon of your nitrogenous base of your guanine. So if we again draw the structure here, this is your 7-methyl group, 7 carbon, in fact it's a nitrogen. You add a methyl group here. So now it becomes guanyl. Okay, right, this CH3 that's added to your guanine is from S-adenosyl methionine. Okay, so you form a 5' cap that's at the 5' end of your messenger RNA, helps in stabilizing the mRNA and helps in translation also, and helps prevent it from degradation. Okay. Now nuclear export of RNA. So because you know the RNA is synthesized in the nucleus, but protein synthesis takes place in the cytoplasm. So this RNA has to come out into the cytoplasm for its translation. So transcription and replication are the processes that occur in the nucleus of a cell, while as your translation occurs in the cytoplasm of the cell. So there's a protein called as CBC, sorry, that's called as a cap binding complex. It's a protein that recognizes the cap, 5' cap of your messenger RNA. It binds with it, okay. And then this complex, your cap and the CBC complex, is recognized by a nuclear pore complex, okay, the nuclear pore complex, and this helps in the transport of this ribosomal, messenger RNA out into your cytoplasm where it's translated.